Hello. Oh. Hold on. Oh my god. Ah. Oh. Yeah. All right. Ah. Welcome, welcome. Come on in. Come on in. It is time for fun. But first, what is this? What are we doing? What is this tomfoolery? The cat's being weird. So this, it's all about microbiology. What is all about microbiology? Great question. So if you haven't haven't heard, or if you've not been around for that long, I have a project I'm working on called All About Bacteria, where the goal is to have a bunch of streams eventually where I just for a whole stream go in depth on a certain bacteria. Because that's very research intensive and takes a while, we haven't had one of those yet. So I decided in the meantime, why not talk about microbiology concepts to kind of hold you guys over and whatnot. So that's what this is. And for our first stream, what are we talking about? Great question. We are talking about gram staining. Let us hop on over to the lovely um, Google Doc. So, this is the Google Doc. I threw this together mostly using information from this virtual lab and what I remember from school. So, welcome. So, firstly, what is gram staining? Why should you care? This is going to come up a lot in the All About Bacteria series once we get there. Gram staining is a laboratory technique that is used to identify the peptidoglycan composition of a bacterial cell wall. Um, and we often use this technique as a beginning, part to identify bacteria. It also has other uses, such as identifying bacterial contamination in food products, such as yogurt, which we'll do in the virtual lab. Don't worry, we'll get there. So, who came up with this technique? This technique was made by Hans Christian Gram, hence the name gram staining, in the 1880s. Originally, it was used to identify bacteria in stained lung tissue sections. Now we use it for different reasons, such as the ones we just went over. So gram-negative bacteria appear pink, which you can see here. Well, they appear pink or red, but this is this is Pseudomonas aeruginosa here in this image. Ooh. You can see here, these are gram-stained bacteria, and as you can tell by the colors, the name. Grammarly, shut the fuck up. You can tell by the colors in the name that they are gram-negative because they are pink. Now, why are they pink? They don't have enough. They have very thin layer of peptidoglycan in their cell wall. And that basically means there's not enough of the peptidoglycan for the violet pigment that is used during gram screening to properly bind to the cell wall. Now, gram-positive bacteria, which you can see here, this is Bacillus anthracis. And if you're wondering what these other cells are, this is from the fucking Grammarly citations in the way. Give me a minute. Give me a minute so I can get the Grammarly citation off my fucking screen. Grammarly, I need you to shut the fuck up. Yes, hello. There we go. There we go. All right, we're back. All right. So, this is Bacillus anthracis, which is what these long string kind of like line shapes are. And this is from a sample of cerebrospinal fluid. And these round objects are white blood cells. Now, white blood cells also accept the crystal violet, sometimes called grand violet stain, which is why they're purple here. They're not gram-negative or gram-positive, they're just blood cells. But because they accept the stain, they are purple. Now, what does it look like when you have both gram-positive and gram-negative cells on the same slide? Here's what this is. So this has gram-negative bacilli. So for bacteria shapes, there's coccus, spirillum, not spirillum, spirochete, yeah, spirochete coccus, which are these little balls, and bacilli, which are rods. So here we have gram-positive cocci-shaped bacteria, which are here, and then we have gram-negative bacilli-shaped bacteria, which are the pink ones. And it's very cool. So that's what they look like. So gram staining uses 4 different chemicals. Grams violet, which is also called crystal violet, grams iodine, decolorant, which is usually 95% alcohol or ethanol, and safranin. So the gram violet will bind to large amounts of peptidoglycan in the gram-positive bacteria. What the gram iodine does is make sure that that binding is kind of stuck in place. So when we do the decolorant step, it doesn't wash away anything but the excess. So the decolorant gets rid of any excess gram violet or gram iodine, for example, on these gram-negative bacteria. Because that gram iodine doesn't really stick to it, it kind of gets rinsed out with that decolorant step, so we can see the color clearly. And then the safranin is used at the end to stain any of those gram-negative bacteria. So what are the steps of gram staining? First, you got to heat fix bacterial samples on the slide. What is heat fixing? So heat fixing is when you take a liquid sample that has bacteria, and to evaporate the liquid and make sure the bacteria, stay still on that slide. It also kills them. You run it over a Bunsen burner heat, and it fixes that bacteria to the slide. So first step is heat fixing the bacterial sample to the slide. And then you go over to a sink, because this is a very messy process. And also you're going to rinse with water, as you can see in the later steps. So you apply grams of violet, and you let that sit for a minute. Then you rinse it, you apply the grams iodine, let it sit for a minute. The reason you let it sit is so that it can have time to do its thing. After a minute, you gently rinse it, and then you apply the colorant. And you only let it sit on the slide for 30 seconds, and then you quickly, but gently, rinse it with the water. And the reason you want to be gentle with this is so that way you don't end up washing off those heat-fixed bacteria. And then you apply the safranin. I can never remember how to pronounce that, I'm sorry. And you let it sit on the slide for 1 minute. Quite literally rinse. I was going to say rinse and repeat, but then after this, you gently blot it dry with bilbus paper, which is a special type of paper. It's very gentle, it's very thin. And then after you've blotted it dry, you analyze it under a microscope. And here are the image sources that we just went over. Gram-positive, gram-negative mixed slides. These were some good images. And now, before we do the virtual lab, do you have any questions? Does anyone in chat have questions about what is going on? Do you want me to go back over the notes we have so far? I know I went through it really fast. I just happen to be very familiar with this topic because I've done gram staining before. I don't think I have images of it, which is unfortunate, but it's very cool. So, before we do the virtual lab, why don't we... Trying to think of what we can do, because I don't want to have this stream be 15 minutes. I don't want this to be a 15-minute stream. Why don't we look at... So this has a good diagram of what gram-positive versus gram-negative bacteria looks like. So the brown stuff is the peptidoglycan. As you can see, gram-positive bacteria have a lot of it, and it's on kind of that outside part. So you have the periplasmic space, and then you have the plasma membrane. So over here, we have the outer membrane, which is made of lipopolysaccharides and proteins. And then you have this very thin layer of peptidoglycan here in the center. You have both a periplasmic space between the peptidoglycan and then between the plasma membrane. So, because here there's a lot of peptidoglycan, it binds really easily. And because here there's not a lot, it doesn't bind as easily. And so that's kind of a nice visual. Thank you, Wikipedia. And then here's a better 3D structure of what a gram-positive cell wall looks like. So you have the plasma membrane, which is made up of the lipid bilayer. So then we can actually see the chemicals that make up the peptidoglycan. Two tetrapeptide chains cross-linked by peptide, N-acylmuramic acid, and then N-acetate glucosamide. I'm sorry. And look, they kind of form nice little rings. That's cute. But that's kind of like a more 3D version. So you can imagine it's a lot thinner on a gram-negative. I wonder if they have an image. Oh, here's like kind of a species, like a good example of how we would identify it. So we know the bacteria is gram-positive, and so then we look at the morphology. And if it's bacilli-shaped, we can easily come to the conclusion that it's one of these 4 groups of bacteria. But if it's cocci shape, we then have to look at how it reacts with certain chemicals. So here, the first step would be looking at how it reacts with catalase. If it's positive, we know it's Staphylococcus. If it's negative, we know it is a Streptococcus. And so if it's Staphylococcus, we would look at coagulase. If it is positive, it's Staphylococcus aureus, and if it's negative, we then look at its novobiocin response. I think novobiocin is a type of antibiotic. Hold on, let me check. Novobiocin. Yeah, it's an antibiotic. Okay, there we go. Back to the stream. All right. And so then we can tell if it's sensitive, it's probably Staphylococcus epidermis, which is just a Staphylococcus species that kind of vibes on your skin. But if it's resistant, we know it's Staphylococcus, Saprophyhacus, Saprophyhacus. So that's what if it's positive for catalase. If it's negative for catalase, we look at its hemolysis, which hemo is blood, lysis is part of how cell walls break open. So if it doesn't do that, I'm assuming that's what this means, then we can tell that it's some type of enterococcus, either Enterococcus facilis or Enterococcus faecum. But if it hemolyzes and the result is this green, we know it's hemolytic A. And then we can look at how it reacts to these type of antibiotics. So sensitive, it's a Streptococcus pneumoniae, but if it's resistant, it's one of these 3 Streptococcus. But if it hemolyzes and its result is hemolysis B, which is clear, we then look at how it responds to bacterin, bactericine, and if it's resistant, it is Staphylococcus agalacinidae, and if it's sensitive, it's Staphylococcus pyogenes. Group A, Group B. So this is Group A strep, this is Group B strep. Yeah, so that shows why it's important for gram staining. Because from this is a good starting point that gives us something to kind of take these webs. Because if we didn't have this knowledge, when we're kind of stuck, because we don't really have a starting point. Because we don't know if it's gram positive or gram negative. But because we know it's gram positive, we can easily go through all these steps to figure out what it is. And if you're wondering what morphology is, it's the shape. We just kind of talked about that earlier. Bacilli are these rod shapes. So Bacillus, Listeria, Clostridium, and then Corynine bacterium. While cocci are those ball shapes. I don't know if you're hearing that, I'm sorry. So these are some exceptions. 1 second. A sibling would like to ask a question. No slurs.
I don't say slurs. First of all, do you like sexy red?
What?
Yes, sure. What are they saying?
Nothing. Also, Snoopy! Ah! Hi, buddy! Get off my keyboard.
Ah. Oh, Bubble.
I have a cat. He is purring. Oh my god. He stepped on my keyboard. Oh god, what have you done? Okay, you can rub your face on it. That's fine. Okay, sorry. Family came home. Everything just kind of got out of hand for a minute. All right. So these are some exceptions that I did not know about because my knowledge of gram staining is very limited so far. It won't be in the future, but for now, it is. So some taxa lack peptidoglycan, and then I give some examples. The Dinococcotia have gram-positive stains, although they are structurally similar to gram-negative bacteria with 2 layers. Okay. The Chloroflexota have a single layer, yet with some exceptions, stain negative. 2 related phyla are also monoderms. So what a monoderm is, it has a single lipid bilayer. And then gram-negative are diderms, which means they have 2. Di means 2, mono means 1. And then here's some information about pathogenicity. In a classical sense, 6 gram-positive genera are typically pathogenic to humans. 2 of these, Streptococcus and Staphylococcus, are cocci. The remaining organisms are bacilli. And they can be subdivided on their ability to form spores. Non-spore formers, which are the Corynelium bacterium and Listeria, and then Bacillus and Clostridium, which are produce spores. And then we divide it again based on their respiration. Facultative, obligative. Yeah. Are 3 gram-positive genera that cause plant disease. So these cause plant disease. These are not people disease. So we got Rathibacter, Lefsonia, and Clavibacter that cause disease. Now, obviously, this talks about gram-positive species, and that because that's the example they used up here. Obviously, gram-negative bacteria such as E. coli, which is gram-negative, can cause illness. Okay, so, and then bacterial trans. Oh wait, this is cool. Sorry. So for context, my senior year, I had to do a kind of like I'm gonna call it a science fair thing, but kind of like research. And originally, what I wanted to do was genetically modify 3 different bacterial species. I went with E. coli. I wanted to do Bacillus cereus. Well, not Bacillus cereus, Lactobacillus enterocephalus. And then I think I did Pseudomonas. Yeah, no, I did Pseudomonas. Pseudomonas florensis, E. coli, and then Lactobacillus enterocephalus. Obviously, some things changed, and originally what I wanted to do was transform them and then look at how much of the protein they output. Eventually, that ended up just becoming how do they actually are they capable of being transformed? So the lactobacillus ended up becoming Bacillus cereus, but everything else was normal. So this is interesting to me. As of 2014, about 80 species of bacteria were known to be capable of transformation, about evenly divided between gram-positive and negative bacteria. There might be no estimates, and several species reported by single papers. Transformation among gram-positive bacteria have been studied medically for the such important species. So medically important just means how significant is this to people, such as Streptococcus pneumoniae, which can cause pneumonia, Streptococcus mutans. Oh, that actually causes tooth decay. Okay. Staphylococcus aureus, which if you don't know, is very bad and can make you really sick. Specifically, MRSA, which is methicillin-resistant Staphylococcus aureus, and then Streptococcus sanguinis, which is a human mouth bacteria. And then gram-positive soil bacteria, Bacillus subtilis. I've handled this one, and I've handled Bacillus cereus. These are cool. I love Bacillus bacteria. And orthogenic notes, I don't. Here. Oh wait, orthographic. All right, so that's some information from Wikipedia. Probably not the smartest of sources, but hey, I don't want the stream to be 15 minutes. So, all right. So, before before we do the virtual lab, let me see how many of you are here. Okay, 2 people. All right. I'm going to drop the link to the virtual lab into the chat. Now, now. I have the audio for this turned off, just so we're clear. There is audio with this virtual lab. So there's the link. Let's go. All right. Let me get my screen up and going. Array. Yippee. Yippee. All right. Oh, wrong screen. Continue. So, hi, lab assistant. Welcome to the yogurt section of dairy processing plant. It appears that our latest batch of yogurt might have been contaminated with some undesirable bacteria. So remember, these are basic rules of lab safety. Wash your hands before and after handling materials. Wear gloves and safety glasses. Don't eat or drink anything in the lab. Be aware of dangling jewelry, clothing, or long hair that will get caught on equipment. Clean equipment after use to avoid possibility of contaminating the next person who uses it. All yogurt has bacteria in it, since yogurt is made by adding specific bacteria to milk. These bacteria are beneficial to humans. Unfortunately, undesirable bacteria like Salmonella or E. coli can end up in yogurt. These bacteria can make people very sick. So this talks a bit about Salmonella and E. coli. Many subspecies of the genus Salmonella can cause subspecies of the genus Salmonella cause infections in humans. For example, Salmonella enteriditis causes foodborne illness. Escherichia coli, E. coli, is commonly found in the lower intestines of warm-blooded animals. Most E. coli strains are harmless, but some, such as E. coli 0157 H7, can cause serious foodborne illness in humans. To help identify bacteria, we look at them under the microscope using a technique called gram staining. This is what plain yogurt looks like. So we have uniform texture, good color, uniform color, and uniform texture. And here is what bad yogurt looks like. You can see there's water on the top. The color isn't very uniform. You can have these air bubbles. There's whey. It's the other whey and water we're separating, yeah. All those air bubbles are forming near the bottom of the bottle. These may be signs of contamination. So this is a photo that tries to better show the air bubbles. I don't think it works very well, but you can kind of see them. And then this is info. Why are air bubbles a sign of contamination? When bacteria grow, they produce gases. Air holes appear at the bottom of yogurt container, indicate presence of undesirable bacteria. The yogurt looks suspicious. But to tell if it is contaminated, we need a much closer look. Let's collect yogurt samples and prepare them for viewing under the microscope by doing a gram stain. So because we're taking a sample of yogurt, what we're going to do is dilute it. I didn't mention this here because it didn't feel very relevant. But gram staining, if you're using something like a sample of yogurt to look for contamination, you're going to have to dilute it. So in the gram staining process, we use different chemicals to stain bacteria that are in yogurt. Then we'll look at them under a microscope. The bacteria will be stained different colors depending on what type they are. And then this just talks more about gram staining. Bacteria that stain a pink or reddish color like this are called gram-negative bacteria. So here we can see their artistic rendition of gram-negative bacteria. By the looks of these, they are bacilli shaped. And their presence. And because the lab is focusing on yogurt, it mentions here that their presence means yogurt is contaminated. So a bacteria that stains pink or reddish colors like this are called gram-negative, and their presence means the yogurt is contaminated. And then here's a photo of what that actually looks like. So this is a microscopic image showing I think it's not cockeye. I think it's bacilli. It could be cockeye in like it's hard to tell. I don't think it's cockeye though. And then bacteria that are used to make yogurt from milk stain purple. They look like this in a microscope and are called gram-positive. Pink bacteria are called gram-negative. You just mentioned that. So these are cockeye. These are dicocci because there's 2 balls that kind of usually form a pair. Some of these are off by themselves, but usually because more often than not, these are pairs of bacteria. Those would be called dicocci for their shape. And here's a photo that does not show what we just saw. These show gram positive, and it looks like they're bacillus. This could just be really bad rendition of bacillus-shaped bacteria, but it does look more like cocci than it does bacillus. All right. We're going to gram stain samples of yogurt so that we can see what kind of bacteria are present. All right, let's prepare our slides for gram staining. So to prevent bacteria from contaminating our work area, we have wiped down the table with a 70% ethanol solution. We will also light a Bunsen burner to sterilize the inoculating loop that we'll use later and help maintain a sterile working environment. So this is a striker. I fucking hate these. I don't like lighting Bunsen burners. Something about it scares me and I feel like I'm gonna get burned every time I do this. So the gas is already turned to low, so I already bring the striker to the Bunsen burner down, flint side facing down, and squeeze the striker to create sparks. Hey, look. So because we're using yogurt, we're going to need to dilute it. So we need to dilute the yogurt before we put it on the microscope slide. We'll use a PBS liquid, phosphate buffered saline, as a diluent to help thin out our yogurt. That helps us see the bacteria under the microscope. And this explains what that is. I'm not going into that. That is pH, and that is for a different time. Start by placing 1 milliliter of diluent into a test tube using a pipette. Hey. This is an inoculating loop. Oh, wait. You can't see the top. A small loop of wire that used to collect samples. So this one is metal. There are disposable inoculating loops. They are kind of yellow and plasticky. And those you don't have to sanitize, really, because they come pre-sanitized. And you can't flame them with the Bunsen burner. But these wire ones you flame with the Bunsen burner to get rid of any bacteria that might be in the surface. We'll use it to place a small amount of yogurt into the test tube with the diluent. So before we do that, we gotta burn it. Also, there are microorganisms in the air, so this, by working close to the Bunsen burner, that gives you less time for those microorganisms in the air to contaminate your sample. It's also, we'll get into aseptic technique in another all about microbiology stream, but today is not that day. So we're gonna sterilize it, and then once it gets that red hot, you pull it out, and you let it cool. That way you don't automatically kill your bacteria. After it is cooled down, use the loop to obtain a small sample of yogurt from the suspect container. Lovely. To get a good sample, reach deep into the container or thoroughly mix the yogurt first. Place the yogurt sample into the previously measured diluent. Now gently shake the test tube for 5 to 10 seconds to thoroughly mix the yogurt sample through the diluent. Use an inoculating loop to transfer a small amount of the sample from the test tube to your prepare slide. And then you sterilize it again. Yes, you always have to sterilize it. No, I don't make the rules. Yes, you should follow the rules. And we're going to do 2 loopfuls. Here they don't sanitize in between loopful, but usually you're supposed to do that. And then we're going to spread it out. Looky. Now we're going to heat fix it. So when... Oh. You saw nothing? I don't know how you're... Why the fuck is that there? I know. So when I said heat fixing, this is what that is. So you see all that water? So it explains it here. Now we'll heat fix the slide. This prevents the bacteria from being washed away when we begin the staining process. So you don't want to overheat it because of denaturation, which changes the shape, and that may lead to faulty staining. So that's why you don't want to heat it too much. And now all that excess water is gone, and you have a nice gray image. So we have 4 stains. Crystal violet is a violet-colored dye. The dye penetrates the cell, gram-positive as well as gram-negative microorganisms. This happens because the component of the dye formally positive charged ions that associate negatively with negatively charged components of the cell wall, aka our beloved peptidoglycan. And now we let that sit for 60 seconds. Now we run it under slow water. And then we do this again. Gram's iodine is a solution that is used in the gram staining process. Iodine interacts with the crystal violet that has associated with the cell's component to make a bigger complex. This complex becomes trapped in the cell membrane. And then here's a visual. Look at that. Don't you just love the precariously named battle? Oh my god. Usually they're not labeled like that. I don't know why they're labeled like that in their photos, but I have some questions. We rinse it again. And now we use this. So it removes it from the cells of gram-negative organisms because of thinner cell membranes, and also it removes any excess crystal violet from gram-positive organisms. So that's why we use this. I wonder how their alcohol bottle is labeled. Oh, they don't show us the alcohol bottle. They just show us them putting it on the slide. Lovely. And then we wait for it for 30 seconds. Because if you let it sit too long, you're gonna get rid of all of it, which is not good. Safranin is a pink colored dye that's stained cells that have been decolorized after alcohol wash. That's why it works, okay? So we drop it. Oh wait, I forgot to look at the photo. And then we wait 60 seconds. And then we rinse it again. And then now that our slide is ready, but it's still wet, so we dry it between bilbous paper. Because it's tightly absorbent. So then we gently press, and then we do this a few times until it is no longer moist. And here is their photo of what a completed gram stain slide looks like. If you're wondering what that black stuff around the edges, that is Sharpie. It sounds weird, but that's a good way of making sure you're putting your stuff in the right place. That's why it's there. All right, let's continue. Now we're doing this our term, on our own. So, chat, which of these comes first? Is it C? Is it G? Is it A? Or is it S? We're having yogurt in the... No, Fabricant, do not eat the yogurt. That's right, chat. The answer is C. Now, how long do we want this to wait? 30 seconds or 60 seconds? That's right, 60 seconds. I feel like Dora, but for microbiology. And then we rinse. All right, chat. Now, what comes next? Grams iodine, 95% alcohol, or safranin? That's right, it's grams iodine. Now, how long do we want it to wait, chat? That's right, 60 seconds. And then we rinse it. Okay, I'm gonna stop bullshitting you and playing Dora. You know what comes next. We do the alcohol. 30 seconds. And then we rinse. And then we do safranin. Drop it. 60 seconds. And then we rinse. So, ignore her. Here is the non-contaminated slide. And as you can see, it only has the desired bacteria. And here is the example yet again. And then here is the contaminated one, which has the undesirable gram-negative bacteria that could make people sick. Here is the example image from before of what gram-negative bacteria looks like. Look at all those gram-negative bacteria. It's a good thing we caught this before the product shipped. Now we need to do some investigating to determine how the yogurt became contaminated and identify the exact strain of bacteria that contaminated the yogurt. The end. Okay. Alright, the end. I did this exact virtual lab in high school. But hello, Fabricant, please do not eat yogurt. Alright. Well, the yogurt in the lab. You can eat yogurt in your house. Alright. That's it. Ta-da. This is the shortest stream I've ever done. Wow. We learned so much, chat. Okay. I'm gonna end stream. Because I only have one person, I'm not gonna raid into anybody. Oh wait, we can raid into Grace. I don't know if rating settings allow for someone to raid into her with only one viewer, but I can't spell. That was a fun all about microbiology stream. Holy fuck, was it short. Although this will be a good YouTube video. All right. What? Why am I? Oh, right. I forgot that Grace's Grace's stream name has fours. So I'm like, huh? Why are there four? Why am I angry? All right. So that was it for the All About Microbiology stream. The VOD for this will be up on YouTube right away. And so yeah, very short stream, but very fun nonetheless. I can't wait to do more of these. I will see you all tomorrow for some art. Yeah, for some art. All right, let's go right into Grace. Have a good night. Remember to stay hydrated. Take your meds. I love you. Goodbye. God, that was such a